Good morning everybody. I am Dr. Nitin Joshi, Professor in Urology with teaching experience of 25 years, working as consultant neurosurgeon in Panvel more than 30 years. Today we will discuss embryology relevant to hypospadias. Sexual differentiation is a stepwise phenomenon where initial step or preceding step determines next step or proceeding step. So, chromosomal sex determines gonadal sex, while gonadal sex in turn determines phenotypic or morphological appearance. The chromosomal sex is determined at the time of fertilization. All human cells contain 46 chromosomes, that is 44 autosomes and 2 sex chromosomes, that is X and Y chromosomes. Zygote being diploid in nature also contains 46 chromosomes. If zygote contains Y sex chromosome, then irrespective of number of other X sex chromosomes, there is development of male phenotype due to development of male gonad, that is testes. So zygote containing Y sex chromosome, then there is development of male gonad, that is testes. So male sex differentiation also depends upon X sex chromosome and 44 autosomes, but Y sex chromosome plays major role in developing male phenotype. Genes located on short arm of chromosome Y chromosome is the main driving force for differentiating indifferent gonad, that is genital ridge, into male gonad, that is testes. This is because genes located on short arm of chromosome Y are responsible for secretion of proteins called testis determining factor, and this region of Y chromosome is called sex determining region of Y, that is SRY, sex determining region of Y. Under influence of these factors, indifferent gonad gets differentiated into male gonad, testes. In 8th week of intrauterine life, fetal testes start secreting male sex hormone testosterone under influence of hormone LH from anterior pituitary of fetus and beta HCG secreted by maternal placenta. This beta HCG also functions like LH. This results into stimulation of interstitial cells of Leydig of fetal testes, causing secretion of male sex hormone testosterone. There is presence of enzyme Y-alpha reductase in genital sinus as well as genital tubercle and genital swelling. This enzyme alpha reductase is responsible for conversion of hormone testosterone into dihydrotestosterone. This dihydrotestosterone, that is DHT, is 10 to 13 times more potent than hormone testosterone. These hormones, testosterone and DHT, are responsible for virilization, and hypospadias means under virilization. Hormone testosterone and dihydrotestosterone bring about virilization through androgen receptor. This androgen receptor belongs to steroid hormone receptor family. Androgen receptor consists of 4 distinct domains, namely amino terminal or regulatory domain, DNA binding domain, hinge region, and carboxy terminal or ligand binding domain. These androgen receptors are present in target organs, that is primordia of external genitalia, namely genital tubercle and genital swelling, urogenital sinus, as well as in the internal ductal system, that is Wolffian duct. Normally, this androgen receptor is present in the cytoplasm of the target organs in unbound state. That is, in absence of hormone testosterone, it is free and stabilized by heat shock proteins. In presence of hormone testosterone, free testosterone diffuses along the concentration gradient from plasma into cytoplasm of cells of target organ. In cytoplasm, entered testosterone binds with the carboxy terminal, that is ligand binding domain of androgen receptor. Due to binding of the testosterone with ligand binding domain, androgen receptor gets dissociated from heat shock proteins, and then it undergoes phosphorylation and then translocation of this androgen receptor with ligand testosterone. Now, this translocation from cytoplasm across the nuclear membrane into the nucleus of the cell. At the level of nuclear membrane, there is presence of enzyme 5-alpha reductase responsible for conversion of hormone testosterone into dihydrotestosterone. This dihydrotestosterone is 10 to 13 times more potent than testosterone. Also, this DHT is having more affinity for androgen receptor than hormone testosterone. So, at the level of ligand binding domain, now DHT binds and there is displacement of the testosterone from the ligand binding domain. Now, this DHT and androgen receptor complex enters into nucleus. In the nucleus, now this complex binds to DNA via DNA binding domain of androgen receptor. The site of DNA where DHT and androgen receptor complex binds to DNA is known as androgen response element. Due to activation of androgen response element, the stimulation of transcription, that is formation of new messenger RNA from DNA. This new messenger RNA will now come out of nucleus across the nuclear membrane into cytoplasm of the cell. In the cytoplasm, this messenger RNA along with transfer RNA and ribosomal RNA bring about protein synthesis in the ribosomes. These proteins thus formed are structural and functional, which bring about virilization, that is masculinization. This process of virilization thus depends upon multiple factors like 1. Secretion of hormone testosterone by fetal testes. This hormone testosterone should be normal in structure and function. Second, there should be presence of specific androgen receptors in the cells of target organ. Third, the androgen receptors should have normal structure and function. Fourth, there should be formation of hormone receptor complex. And fifth, post hormone receptor complex, there should be the synthesis of proteins which should be normal in structure and function. Whenever there is any block or abnormality or hindrance in any of these steps, this will result into inadequate virilization. Normally, during process of virilization, there is virilization of the primordia of external genitalia as well as primordia of internal ductal structures, that is Wolffian duct, as well as virilization of the primitive urinary tissue, that is definitive urogenital sinus. The lower urinary tract, that is bladder and urethra, develops from primitive urogenital sinus. Due to draining of Wolffian duct into primitive urogenital sinus, it gets differentiated into proximal vesico-urethral canal and distal definitive urogenital sinus. Irrespective of presence or absence of hormone testosterone, vesico-urethral canal develops into urinary bladder and proximal urethra. So, in males, prostatic urethra, which is proximal to veru, develops from vesico-urethral canal. The prostatic urethra, which is distal to veru and membranous urethra, develops from pelvic part of definitive urogenital sinus. This distal part of the prostatic urethra and membranous urethra develops even in absence of hormone testosterone. In female, this phallic part of definitive urogenital sinus is responsible for development of distal part of urethra and vagina. In presence of hormone testosterone, there is virilization of the primordia of external genitalia, that is genital tubercle. It elongates to form phallus. Mesenchyme present in the genital tubercle condense to form cavernous tissue. The phallic part of the definitive urogenital sinus develops properly only in presence of testosterone into the bulbar urethra and pendulous urethra. Since this urethra, that is phallic part of the urethra, is related to penile tissue. When under influence of hormone testosterone, genital tubercle elongates to form phallus. Simultaneously, under the influence of hormone testosterone, the phallic part of definitive urogenital sinus develops urethral plate. The ventrum of the genital tubercle shows superficial and deep urethral grooves. These superficial and deep urethral grooves are precursors of anterior urethra. These urethral grooves unite to form urethral plate. These urethral plates elongates along the ventral aspect of the elongating genital tubercle. So, there is a simultaneous elongation of the genital tubercle to form phallus and simultaneous elongation of urethral plate along the ventral aspect of the forming phallus to form the anterior urethra. Tubularization of urethral plate results into formation of urethral tube, that is anterior urethra. Due to inadequate virilization, there can be various congenital abnormalities. 1. Improper virilization of genital tubercle, so no proper elongation of phallus, so there can be micropenis. Inadequate virilization of phallic part of definitive urogenital sinus will result into congenital short urethra causing ventral chordae. Improper virilization of phallus will result into, along with improper virilization of urogenital sinus, is responsible for typically hypospadias. Deficiency of penile tissue due to inadequate virilization of phallus. Then deficiency of urethra due to inadequate virilization of urogenital sinus, and along with that deficiency of the skin and associated fascia, that is dartos and the buccal fascia, is responsible for typical hypospadia presentation. Due to undervirilization of the genital tubercle, there is deficiency of penile tissue, that is deficiency of two corpora cavernosa and corpus spongiosum on the ventral aspect. So there is deficiency of corpora cavernosa on ventral aspect, that is there will be unequal corpora cavernosa and unequal tunica albuginea covering corpora cavernosa. That is, the corpora cavernosa on the dorsal aspect will be longer due to deficiency of corpora cavernosa and tunica albuginea on ventral aspect. This inequality of the corpora cavernosa on the dorsal aspect and the ventral aspect can be corrected by TAP, that is tunica albuginea application, during correction of hypospadias, that is the stepwise release of chordae, provided penis is of adequate size. Also, there is deficiency of the other penile tissue, that is corpus spongiosum, which harbors the anterior part of the urethra. Since there is no support of urethra to penile tissue from ventral aspect, there will be ventral curvature, that is chordae. Also, with deficiency of corpus spongiosum and anterior urethra, there is deficiency of covering skin and fascia, namely dartos fascia and buccal fascia on the ventral aspect. These all absent tissues are replaced in hypospadias by fibrotic dysgenetic tissue causing chordae. Presence of normal prepuce on the dorsal aspect with the absence of this skin on the ventral aspect is responsible for typical dorsal hood. Thank you very much.